Okay, I'm Linda Peacock and I'm grower liaison and technical specialist for KVH at the moment. And Steve Hoyte is from Ruakura, and he's one of our research scientists, which is a long-term association with the work that has gone into PSA. So similar to what Desi said, nothing stands still, and we're very, very aware that PSA doesn't stand still. I mean, it's 2010, we had our first incursion. PSA is pretty widespread throughout New Zealand now, not the South Island, and growers are managing it as business as usual. However, a kiwi fruit vine health is not sitting out on the back couch smoking cigars. We're still out there very, very in touch with what PSA might be doing. The National Kiwi Fruit Pathway Management Plan specifically recognises PSA as a high-risk organism, and kiwi fruit vine health is still responsible for making sure that we don't get things popping up like new PSA variants or resistant variants spreading through our industry. So annually, the industry does do a survey looking for resistance to control products, and then every 2 years, I'll let you in on the action, every 2 years we also look at any genetic changes, because we know that it's a biological organism, so it is going to change. So last year, Plant and Food, as part of their normal survey, doing that genetic survey, they actually detected a new PSA variant. They sequenced it, and then they checked how it behaved against the wild type on both Hayward and Hoyte 16A. Now, what they found is that that new variant outcompeted our common or garden PSA when it was put on the Hoyte 16A. So it was like, okay, we might be giving KVH a little bit of a call about this. So they got in touch with KVH and Zespri, and then we needed to work forward to understand what that might mean. So the team got together and started working out what were the things that we needed to know. So first of all, we needed a new PSA test to pick up the variant reliably. We wanted a sampling strategy to check out the variant persistence and spread on the orchards. There was 2 orchards at this point where we had identified that new variant within a sample. We needed to go out and do some leaf sampling. And we also had 300 PSA isolates left over from our resistance testing that year, so why not screen those? So they were from a range of different growing regions, so that would give us another opportunity to see whether it might be spread. We also needed to now know, so how does that new variant behave when you put it on Gold 3, and how does it behave when you put it on Red 19? So, and we also had to think about how would we detect enough PSA out of the orchard so that we can do that work. So KVH went out and started sampling the 2 orchards where that variant had been found. The grower communications were that they hadn't actually seen much PSA on the site that year, which was a good thing. And on the other side of the fence, though, it was late in the season. Rain held us up, no surprises, and the leaf canopy was already yellowing and the leaves were breaking down, so we had a bit of a tough job trying to find some samples. One of the orchards had already been pruned, so again that limited what we could find. However, we did find some leaf samples, and those PSA cultures were tested. We didn't find the variant in that, but we knew, we couldn't stop there. We needed to go out and sample again the following year. And at that point in time, we did work with the orchard owners, and a formal notice of direction was put on the property. That was just to make sure there was no potential movement of risk off that orchard. I mean, at this time, we didn't know if it was a risk, but we needed to kind of freeze the scene. In spring, we went back out again, and we had a great sampling strategy devised by Plant and Food. Just a note, we knew exactly which bay those original samples had come from, fantastic traceability. And again, we went out and looked. Again, as research usually works, there was very few PSA symptoms. We do know that leaf spotting on gold varieties is not as prevalent as on Hayward, so we just knew that already. So then we thought about the fact that we could use the trap plant methodology that we'd used in our previous research, and Steve will talk more to the detail of that. So that was to improve the sensitivity of detecting it within those orchards. In spring, we also did some delimiting surveys. So KVH went out and about, not only sampling those 2 original orchards, but we kind of drew some rings around orchards. That's a mock-up map, by the way, so don't try to work out whether it's your property. The red dots indicating close orchards, which we also monitored, and then further afield there's some orange pins, and what they represent is orchards that are associated with that original PSA variant orchard. They're associated either by people movements or plant and machinery movements. So either people are moving between those orchards or equipment and machinery, and I'm sure they will be logged from this day forward on the technology that we have been talking about recently. So we looked over 25 orchards, and those samples were processed through Hill's lab. The results of those told us that we had picked up 4 additional orchards which also had that variant. So now we had 2 regions with the variants and a total of 6 orchards. So we still need to know from the science whether that was actually a problem. In Hill Laboratories also by this time had checked out the 300 isolates that they had been given, weaving in and out of the COVID testing that they were also doing at that time. And what we found from those samples is there was no additional variance from those regional samples, so that was a good thing. So now I'll hand over to Steve, and he'll talk to you about the science from the start to the end, and then I'll just call back in and say thanks.
Thanks, Linda, and thanks for the opportunity to talk about this project. So yeah, as Linda mentioned, there was a new PSA variant was discovered last year and reported. This graph on the right here shows you the initial data that came out of Mount Albert genetics team, where they, after identifying that there was a genetic difference in the PSA strain, they tested them on small tissue culture plants inside little plastic tubs. So it's the easiest way to test PSA infection. And on the left-hand side there is the 2 little bars there shows you that you're basically putting equal amounts of the wild-type PSA and the new PSA variant onto the plants at the same time. And then the bar in the middle, the plot in the middle there, shows the outcome that they got for Hoyte 16A, where the wild-type, which is in the blue bar, outcompeted the variant type outcompeted the wild-type quite significantly. So there's a lot more population growth of the variant on that cultivar. But you can see on the Hayward there on the right that there was no difference between them after the plants have been incubated. So you got a response on Hoyte 16A, which, as you all know, is very susceptible, and no response on the Hayward. So the obvious questions were, what's going on with these other cultivars that are out in the industry? What might happen on a potted plant, or what might happen out in the orchard? And as Linda's been alluding to, how widespread might this variant be? So the project got underway last winter. So just showing you, Linda's mentioned some of these aspects, but just sort of trying to show you some of the linkages between the different projects that had to get underway to try and get this project going urgently. So the genetics team up in Mount Albert had to rapidly develop a qPCR method so that they could reliably identify this new strain. That information was passed on to Hill Labs so they could carry out the rapid screening. KVH, as Linda said, went out and did a whole lot of extra sampling out in the orchards to try and look at the persistence and any spread that was going on. And the team up in Auckland also wanted to launch into doing more of these competition assays on Gold 3 and also on Red 19. The Gold 3 plants is easy to get hold of. We get them from Multiflora regularly, but Red 19 are not available as tissue culture plants, so we had to contact the team down in Palmerston North who look after the repository of all the kiwi fruit for the planted food, and it's about a 6-month process for them to bulk up enough Red 19 plants for us to be able to use in this project. Following on from that, as Linda mentioned, the sampling strategy was passed on to KVH so that they could have some reliable statistical ways of going into the orchards that were of interest, where the strain had been found. We started using trap plants out in the orchards in the spring last year to try and get increased recovery of the PSA from the sites where it was difficult to find the PSA. And we launched into a set of plant assays, so growing potted plants up at the glasshouse, and using our facilities at Ruakura, we're able to inoculate PSA of the wild type and of the new variant strain onto potted plants of Gold 3 and Red 19, and actually quantify what the response was on those plants to see if it was actually more aggressive or not. This shows some of the competition assay data. So the Hoyte 16A data here on the left is the same as what you saw earlier. The colours are changed. So the green is now the wild type variant. The purple is the new PSA variant. And over on the right-hand side, you've got the result that we saw before with the Hayward. So you can see the result we've got for Gold 3 here. There's no difference between the wild type and the PSA variant. However, on Red 19, there is a slight difference. There's a slightly more PSA resulting from the PSA variant than there is on the wild type. So Gold 3 looked fairly good and a bit of a response on the Red 19, but it was to a much lesser extent than what we see on the Hoyte 16A. So the next question was, what happens when you put these two PSA strains onto potted plants? And you can see the little diagram up on the side, the picture up on the side there, that's the scale of these assays. It's just on the tissue culture plants in a plastic tub. So we set up a series of pathogenicity testing assays at Ruakura, looking at the wild type and looking at the PSA variant strains. We had 2 wild type strains of PSA from Joelle's lab and 2 strains of the PSA variant that had been discovered. We were working on both Gold 3 and Red 19, and we did a total of 9 assays across both of those genotypes. We've got 2 main ways that we can inoculate PSA onto plants. Firstly is to just spray the underside of the leaf with a suspension of the PSA, incubate it for 2 or 3 weeks, and you'll get leaf spotting necrosis develop. We can also inoculate directly into the stem with this little tool that we developed at Ruakura a long while back when we first started our PSA work, and that puts the bacteria directly into the stem, and you get a systemic infection that gives you a lesion on the stem, but it can also lead to systemic leaf necrosis, wilting of the leaves and dieback of the stem and so forth. And all that happens within a matter of weeks. We put the plants generally into high humidity tents for 3 or 4 days and then open them up to give them more normal conditions in the glasshouse, and then score them after about 3 weeks. So this shows an example of some of the results. This is from a Gold 3 plants the leaves were inoculated with PSA. So the 2 graphs on the left, the 2 bars on the left-hand side here is our wild type PSA, 2 bars on the right are the PSA variant examples. And you can see this is the area of leaf necrosis, so it's the percentage area of the leaf that's got necrosis after 3 weeks. And there's no significant difference between the individual strains, and more importantly, the average of the wild type strains and the average of the 2 PSA variant strains is basically the same. So no significant difference in infection of the leaves, and there's other assays that basically gave the same result. This is an example of a Red 19 experiment where we're now inoculating into the stem of the plant, and this shows you the length of the lesion that's developed after 3 weeks on the plants. Same setup, wild type on the left, PSA variant on the right. We're seeing a little bit of variation between the individual strains, which you'd expect, but no significant difference between the PSA variant and the wild type PSA, which is a really good outcome. So just following up on Linda's mention of us using trap plants, we'd used this process a lot in the Gold Futures project, which ran for 5 years, where we were looking at management of PSA in Gold 3 orchards, and we monitored PSA populations out in the orchard using trap plants, Hayward trap plants, to monitor the PSA populations 3 to 5 times over about 5 years as a way of tracking the PSA in these orchards we were studying. So the process is to we were using, in this particular trial, we were using either Bruno seedling plants, which we had available at the time, or Hayward tissue culture plants, growing in pots. We suspend them in a bucket out in the orchard underneath the leader, so they're out of the way of the tractors. They stay out there for 1 to 2 weeks. We've got a water supply inside the bucket so they don't dry out, so they don't need to be looked after. And we try and target a time period when there's going to be a rainfall event, so there's going to be some dispersion of the PSA in the canopy. So we were going out to these 2 orchards of interest that Linda mentioned, and there was very little PSA out there. KVH were going out doing repeat sampling and couldn't find much PSA at all. We put 20 trap plants out into each orchard. One orchard we went into with 2 rounds of trap plants. You can see on the right-hand side there, it's a little bit hard to see, but when I was putting the plants out, I found an infected shoot up in the canopy and put a plant underneath it, and not surprisingly enough, that was the plant that had the most spots on later on. So we collect the plant, they're out in the orchard for 1 to 2 weeks, bring them back into a dry covered area so that there's no more rain splash going onto them, so that whatever develops on there is what's come out of the orchard. They're incubated there for 2 to 3 weeks. You get leaf spotting developing to some extent. KVH came along, we were able to sample these leaves, send them off to hills, and with the new qPCR they were to tell whether the variant was present or not. And there's a gel result there that shows the example that the PSA variant has got a gene that's missing, the wild type has got that gene present. This is just a summary of the results from all the isolations and screening that's gone on. So the first 2 rows there show the samples that were taken from the trap plants at the 2 orchards. We've got a lot of isolations from the site A, but only 27 of them came up positive for PSA, and of those, none of them were the PSA variant. So that showed us that the frequency was low, which matched up with our previous understanding of that orchard, but it doesn't mean it's not there, it just means that from that small sample, we haven't picked it up. Whereas at the site B, we only had a small number of samples, 6 samples that they were able to isolate, and all of those samples gave us PSA, and each of those isolations was the PSA variant. So we got 100% recovery, but it was a very small sample, but it was able to confirm that the variant was still present in that orchard. And the line here, the site A and B and all the other sites, that's encompassing all the trap plant data, all the isolations that were taken from the copper resistance survey, and all the samples that were taken from the neighbouring orchards and connected orchards. So we had less than 50% of those were giving us a PSA positive result, and there were only a total of 11 PSA variants were collected from those, 6 of which came from the trap plants. So we only got a 4.8% from those. So the average overall was only about 6 or 7% recovery of the PSA variant. So it's still out there in these orchards, but from what we've seen from all the potted plant assays we've done, that the variant is not it's not more aggressive than the wild type, so there's likely to be no concern, but it's obviously something that needs to be monitored going forwards. I'll hand it back to Linda to finish off. Thank you.
Thanks, Steve. Okay, so at the end of all that little piece of excitement for the year, in terms of PSA variants, we found 6 orchards represented across 2 regions. Interestingly, and I think importantly, both the grower and our field work showed that there wasn't kind of runaway PSA happening on those orchards, but it was still important that we had followed up on that. The potted plant assays on Gold 3 and Gold 19 showed us that there was no difference in the symptoms between the wild type, and also there didn't seem to be any, in this case, disease challenges associated with it. So with that information all gathered, KVH and Zespri concluded that there should be no negative consequences from that variant. So we were able to then go back to the orchards where we had placed those nods or notices of directions and just free up the movement on and off those orchards because there just was no risk associated. So what that investigation did show us is the huge value of the collaboration that we've had across the years at a research level and just at a personal level, working with a number of providers and also our kiwifruit community. As usual, the growers are asked going forward, if you do feel that you see any unusual symptom going on in your kiwifruit, either at PSA or other things, please be in touch. I mean, in previous years around the PSA space, we had actually investigated a couple of orchards where they felt that they had a bit of runaway PSA, but we, in that case, were able to, with the backup of plant and food work, find out that actually there was no resistance, there was no change in the genetics. It was just kind of a bad year. So there's always that opportunity for us to clarify that. Going forward, also the genetic monitoring of PSA will continue because, again, it's a biological organism. It will continue to change. Lastly, I would really like to thank the growers and orchard managers. They were tremendous in helping us in this study, and I think that that's largely because they understood the big picture, which is so important. Thanks also to Zesbury Innovation Team for the budget and also for the help in the response planning. And Plant and Food and Hill Laboratories, because they actually prioritise this research for the orchards in our industry when they're actually super busy people. I haven't got time to go through individually all of those people there, but those few short lines represents a lot of hours, a lot of late nights, a lot of work under pressure, and a lot of desire to find the answers for the industry. So, and that's it, thanks. In this case, Houston was able to stand down.
Don't run away, don't run away. We're just, any questions out there? Oh, crikey, you've opened a can of worms. Here we go. I'm going to get off the stage. You two get up here.
Thanks both. This work originated out of the original resistance work. Was there any specific work done around this variant on its susceptibility to treatment?
There was some work done up at Mount Albert, as far as I know, and there was no difference in response to control products. Yeah.
Great work, Linda and Steve. My question a little bit outside of the box. So we're looking at variants that are going to be more virulent. What about looking for variants that outcompete the wild type that are less virulent, that we might actually want to use and spread around, vaccinate, inverted commas? So are we needing to actually widen our scope here and look at the opportunities that some of these variants may provide kiwifruit growers in the future?
I can see Juliet Ansell in the audience, and she would be far better placed to talk about this. However, what I can say is that certainly we have in a number of the research projects within the PSA steering group, which now sits under the Innovation Crop Protection space. There's exactly that work going on, Barry. There's a PhD thesis going on around looking at what else is living out in amongst our environment in that PSA space, to see that it might be used as exactly that, inoculum, or built through to control products. I think in total we've got at least 2 or 3 projects sitting in that space already. We've also had a look, Joelle Vanest was involved in a program looking at microbiomes, because we know that's a whole new area of expertise. 20, 30 years on, we'll know a lot about more in that space. So that's been a very forward-looking, blue sky thinking research portfolio. So there are a number of things that haven't quite come to the surface, but a lot of information has been collected, and we've got a lot of leads, and all we need is a bit of cash, and away we go.
Me again. Excuse me. Really interested in those trap plants. When you first mentioned them, I thought you might be using Hoyte 16A because it's super susceptible. So why do you use Hayward and Bruno?
Yeah, well, it comes down to how easy they infect leaves. Actually, leaves of Hoyte 16A are not particularly susceptible to PSA, so the Hayward is the best plant to use. And Bruno's, as you know from your rootstocks, growing out from your base of your plants, is quite susceptible to spotting as well. So, yeah, it's all about the susceptibility on the leaves and getting easy symptom expression. It's
just a slow development photo of what's going on. It's really what it serves us.
I was actually just wondering, does the act of cultivation in orchards make the PSA more likely to mutate, or is it just as likely to happen in wild populations, like in the bush?
And fortunately, once again, and you assisted with this project, Jamie, so we have collected samples from both wild kiwi fruit and cultivated kiwi fruit on the borders of the gullies. And that's some of the work that we've looked at around microbiomes of both the plant and the roots. And we've also looked to see if there's any variance in the PSA. But in fact, the wild kiwi fruit showed lesser incidence to PSA, so potentially there's something that we're doing in that monoculture environment which is allowing it to kind of sneak through the gaps, but it's certainly on our radar as well. Jamie, thanks for the question.